Welcome to the Diary of a Medicinal Chemist.
So in the drug discovery stage, we have mentioned that it is also important that we begin by identifying an appropriate biological target. Now, what that simply means is we are trying to identify the specific macromolecule. It could be a macromolecule, for instance, a protein, which could be an enzyme, could be a receptor, could be a ion transport channel, could even be DNA, could be DNA, which is important. In other words, which the drug we are trying to design would act upon. So there are so many methods that are used in target identification and validation. Now, one approach to identify targets related to a disease is to uh, compare the genetic makeup of a large number of patients living with the disease with a large number of normal patients, and then you kind of identify which genes are consistently different in the two sets. Now, given that there are over 20,500 genes in the human genome, there are many sites for mutation that, of course, can lead to a disease. But however, we have less than 10% of the human gene that has been explicitly associated with a disease. Now, another method is to employ one of the several methods of selectively eliminating the function of a particular protein and observing the consequence in an isolated biochemical pathway or even the whole organism. Now, what this means is that there are several methods through which we can kind of knock out a particular protein, then observe the effect of that knockout on the overall physiology of that animal. So that again will give us an indication of the importance of that particular protein as a target. The most commonest approach is to use a target that has already been validated in the clinic. Now, what we mean is already we have for many diseases, we already have drugs developed for them. So what we need to do is to look at those targets that have been used in the development of those drugs. So we would also simply toe that line by also designing drugs that would also act on that same target. In this case, drugs that are gotten from these processes are called me-too drugs. Drugs that act by the same mechanism of action and are structurally very similar. And we have a lot of them in the world today. Now, the process of target identification has become fairly interesting with a number of computer-aided drug design methods. We now have the structural genomics, the functional genomics, proteomics coming on board, and so many other methods. Particularly when you look at the PCR, polymerase chain reaction. Through that, we can do a number of gene sequencing and so on. So it has become pretty much easier, and even predicting targets computationally with the use of computers. That has also given the medicinal chemists, and drug designers another horizon of hope in terms of identification of targets. So afterwards, we have the next thing we have the lead identification, identification and optimization.